Okay, so welcome to the statistics seminar today. It's my great pleasure to introduce Vera from Department of Biostatistics, University of Michigan, who I was very fortunate to work with as a PhD student at Rice. Broadly, Vera's research interests are mainly in high-dimensional data modeling and Bayesian inference. More specifically, Vera and his research team work on functional data analysis, graphic models, Bayesian semi-parametric, non-parametric models, and Bayesian machine learning. So these methods are motivated by large biomedical data sets, such as multi-omics data and high-resolution image data. Today, Vera is going to tell us about his work on Bayesian integrative approach to enable precision medicine. Let's welcome our speaker, Vera.
Thanks a lot, Yang, for the generous introduction. And so as Yang mentioned, I'll be talking about a few things that, a few problems in precision medicine that actually has consumed me for the last couple of years, as I've been working in a biostat department, engaged with with the academic hospital, And so sort of the stream of the thought here is, how to do data integration and more concretely, how to use that approaches to enable, the implementation of the precision medicine. Now, I'll make that clearer, in some more specificity as I'm going through the talk here, right? Let's see. Okay, so I want to start with this. I was trying to dial back, what was my first ever seminar, my last talk at Texas A&M. And it turns out my last talk was my first ever seminar I ever gave. And if you see here, it was back in 2004. And this was my practice job talk that I had given when Bunny and Ray told me, you should practice and you can use one of the seminar slots. And I was petrified, to be honest. When I gave that talk, and it was back in room 150 blocker. I don't know if it's the same room, the seminars are still there, but it would have been nice to come in person and revisit that. But anyways, sometime hopefully down the future now. And back then also I used to talk about Bayesian methods in bioinformatics, and I haven't veered off that track, for a while now, 16 years later. So brings back some good memories of the department, where basically I grew up now. So I'll start with a higher level overview of even a 70,000 foot or even 100,000 foot overview of what precision medicine, putting it, contextualizing this. Now, what sort of problems, and before going into more specific pieces of the problem that we are trying to tackle, within my group as well as with my collaborations here. So there's this whole scheme of P4 medicine that sort of has come about in the last few years or so, I should say, which is basically the 4 levels where there is the predictive medicine, which is more on the risk identification. There's preventive medicine, which is how to avoid, reduce, or monitor risk. And more pertinent to this talk would be the concept of personalized precision or stratified medicine. There are different nomenclatures to it, where you can actually do clinical interventions based on different sorts of data, where most of it comes from either genetic or molecular or some characteristics of the patient. So this is after the fact that the disease has developed. And there's this more broader concept of participatory medicine, which sort of engages at a more social level, where patients themselves become a stakeholder in their health management. And this is sort of stemmed by not only advances in clinical procedures, but also that we collect the data on them. Okay. And that, it's nice to have this demarcation of what exactly levels of precision, personalized, stratified, preventive that we are working in. So just to drill down a little bit, this is the context. And oops. right, hang on 1 second. My mouse isn't working. There you go. And so, so when I graduated from Texas A&M, 15 odd years ago, when I walked into MD Anderson as a faculty member, that was right around the time when the TCGA project was happening in a cancer center. And I still remember walking into a colleague of mine, Keith Bagley's office, who was telling me about the TCGA project. And that sort of, that's exactly what the expression I had on my face when I saw that opportunity, in working with different kinds of molecular data, in particular. But what's and to be honest, like 15 years hence, I'm still, feel there are a lot of unsolved problems and more solves problems as well as opportunities in this area, which some of it hopefully will make sense. And just for the the students, trainees in the group, this is a very a rich area of investigation with multiple opportunities. So with the promise comes the challenge, that the routine technologies now cover such a vast array of data that not only includes different omic data, but also that that one can in almost close to real time measure different kinds of biosensors data that includes different kinds of activity level data and even the social milieu and also different kind of imaging. And that sort of besets the question is, how do we extract biologically or clinically implementable knowledge from these data sets? Okay, and it won't be an understatement to say that the integration, the data integration, may be perhaps the key quantitative challenge in driving modern science or modern biomedical science in that respect, right? So I'll drill down more into the cancer context. It's a disease system that I've worked for a few years now, and I've gained a little bit of, by no means an expertise, but at least some understanding. So there are many bad things that happen with cancer, but one of the good things is that it's actually one of the most well-characterized pathophysiological or biological systems at different data levels, especially at the different molecular levels. By molecular, I mean here genomics levels. And this is actually spurned through, as I mentioned before, that multiple types of high throughput data that are now available on not only different model systems, not only on patients, but also different kind of model systems that include cell lines, which are laboratory-based systems, patient-derived xenographs, organoids, which is basically virtual organs that are created in the laboratory. Okay, and these have been spurned by not only by international consortium level effects that includes the Cancer Genome Atlas, the ICGC, all of which is part of the NIH Genomic Data Commons, but also at a higher level, at federal levels or at consortium levels. But at the same time, even like institution specific, now many institutions have these sort of data repositories that are growing actually day by day. And a couple of examples local in Michigan is our Precision Health Initiative. And within it, we have a Michigan Genomics Initiative that's collecting actually a very neat set of biobank data that not only includes genomic data, but also many kinds of electronic health records data that can spurn a lot of questions that can be asked of it, right? So within cancer, to define what precision oncology means, in terms of doing it within the context of cancer, so I found this nice delineation, that's sort of a standard in the literature sort of right now is what is precision oncology. So 5-10 years ago, this concept came into understanding, or folks started to recognize that you could do much better than treating populations, but going more at an individual level, and that included, the 1.0 version of that would be, just having a small number of molecular abnormalities, some choice number of genes, and looking at always constrained by the tissue of origin. What that means is by whatever cancer type it is. Is it lung cancer? Is it breast cancer? Is it brain cancer? Right. And sort of the current state of the art is what is usually called the precision oncology 2.0, is that the small number of molecular abnormalities has been increased to a few more, dozens or hundreds of these possible mutational hotspots or known cancer associated genes. And then going beyond the organ side, beyond tissue agnostic, which is going and treating the actual genomic aberration or the molecular aberration rather than the actual organ, right? And I found this nice review article that lays out now, at least in some conceptual format, what might be the future. Is a precision oncology 3.0, where this is sort of a life cycle of research and treatment, where the patient is treated, there's monitoring, then you take out different kinds of data from them, and that actually feeds back into a system that can do much more advanced analysis, and then feeds back. So it's sort of like a bench to bedside, and back, and that involves multiple levels of not only data collection, but more refined analysis at pan-omic levels, which is multiple genomic levels, and finding different regulatory elements that drive different cancers together or individually, right? So my talk today there was sort of a high-level summary. So in terms of contextualizing the specific problems from these that we are sort of attacking is how to integrate data from not only patients, but also different model systems. So that goes more into drug treatment and pharmacogenomics. That's what's usually called. And it's going actually completing the circle from discovery to translation. It's the discovery space of things of finding molecular markers. That's a really nice and comfortable area to sit in. But how do we get beyond that to actually make it translatable? And that's perhaps qualifies what I mean by enabling precision medicine. How can as statisticians or more generally data scientists, we can do that, right? And so that'll be the bulk of the talk. And if I have time, I'll just hit upon another recent project of us is how do we get into regulatory mechanisms of developing network-based models for panomic data integration, right? So on the first part of this integrating data across model systems, so just a brief bit of background where this schematic sort of represents like what is the current state of medicine, where you have cancer patients with a specific set of, for say example, colon cancer, and how first they are given therapy and then divided into subgroup analysis and figuring out the effects. So what generally is sort of accepted and what should be the norm in the future, hopefully, is to use the data from existing patients so that we can demarcate patient into subgroups defined by different levels of therapy, and then find the therapeutic effect of these sort of different therapies. So the high-level goals here is basically we have patients from different populations. We have basically samples from different populations. That includes patient-level populations, say from different consortium level. We have cell lines that have been studied in the laboratory, which are basically systems that can be manipulated in the laboratory and studied for drug effects. And there has been a large-scale effort in defining these cell lines, calibrating them, and different drug responses. And sort of the high-level goals is, how do we, integrate this data? First of all, to exploit the conserved biology between these model systems, and more pertinently, use that biology to calibrate what might be a therapeutic index, therapeutic response of these drugs in future patients, right? And at the same time, find optimal sort of the cell line avatars, which is basically, for a patient, is there a set of model systems that are most representative of these patient tumors that can act as these proxies? And finally, what drives them? What are the key genomic drivers that explain these cell line similarities? And it turns out actually, this idea actually grew out from a conversation with one of my collaborators back when I was in MD Anderson, So this inherently becomes a quantitative problem, and the quantitative problem, the way we attack it is like, how do we define joint probability models across patients, cell line, and drugs, right? To achieve these goals, okay? And that's sort of our task here, right? And I'll tell you just, one specific instance of how this can be achieved. There could be many more approaches that are out there. And the literature is sparse in doing this, but still there's a lot of room for improvement. So digging into what is the data structure type, what is the conceptual exact problem that we are trying to attack here, is think about, we have a p-dimensional variable. So think about genes or proteins or any genomic feature, a high dimensional feature from cell lines, okay? And we have n of those. So the cell lines are these are cell lines. These are replicates that have been assessed in the laboratory, and on them we have drug sensitivity data. Basically, because you can test them out in the lab, you can give different sets of drugs. So Q is the number of drugs here, and N is the number of cell lines that these drugs have been tested, right? And then, and then we have a big data set of patient-specific genomic features, where the genomic features are sort of matched up. So these are the same set of p variables that we have on the patients, and it comes from a different population, from a different data collection procedure. And we have, say, M patients, so where M could be potentially large. And the key idea here is, how do we get the drug response? So these patients are not part of any clinical trial, so to speak. So this is just their prediction. What we want to do is integrate these two data sets to find out what might be their drug response, and using the shared features between the cell lines and the genomic features, right? So in that essence, this, if you think about it as a prediction problem, this is our training model system that we have data on, and this is our target model that we want to get to do this, right? So to get to the drug prediction, essentially, right? So what would be a naive way to do it? Now, a naive approach would be to just train a model on the cell lines and the drug sensitivity. Okay, you get a training model from them, and then you can plug in these features and plug into the training model for the prediction. So you can do it independently on the two data sets as a training and test data sets. All right. But what this might not do is potentially account for the genomic similarity that might exist because of this genomic similarity or difference, because the key assumption here is that cell lines will capture all the characteristics of the tumor in patients, which is a huge assumption to make. So if there is some shared variability, that should help you. But if there's no shared variability, that won't help you in the prediction. Okay, and how to do that in a statistical manner? That's basically the essence of the problem here. So what we thought about this is, think about it as a calibration problem, right? So there could be multiple ways to do this. Like I said, like this is one possible approach. Is having a supervised model, a cell line drug model between these two, because you have a set of covariates, you have replicates, and you have the responses, and then you have a set of genomic data sets from two populations, and have a cell line patient model which is more unsupervised, and then tie the two together to predict the induced drug patient model. And that is sort of a semi-supervised approach, which basically combines a supervised and an unsupervised approach. Okay, and basically that's how we combine that, and that's sort of the intuitive idea behind the model building, right? Into getting basically this piece here, which is the drug response prediction. All right. So we call this the Individualized Therapeutic Index Model, the Irx model. And this work was actually spearheaded by a former postdoc of mine, Abhishek Saha, who used to work with me at MD Anderson, and he's now a fellow at NIH. And Minjin Ha, who was my former postdoc and now an assistant professor at MD Anderson, who's, this contributed a lot to this work, right? So getting into the exact formulation of the model, okay? So we have a set of data. In the supervised approach, we're gonna call that labeled data. So we have the cell lines, call it CJ, where J goes from 1 to NC. NC is the number of cell lines, and DJ is the drug responses that is known on these. So for now, assume it's continuous drug responses. So there's a, now on these cell lines, I should have said that. So the way the drug responses are evaluated is you collect a bunch of cell lines, or basically they're a set of cells, you give them a certain drug, and you see how many cells die, basically. No, and at what concentration, how many cells die. So that tells you how potent the drug is, which is called sensitivity, or if the cells don't die, how resistant it is. So because it might not. And these cell lines are tumor cells from tumor cells that are obtained in a petri dish. So it basically gives you some measure of drug activity. So you can think about higher DJ having higher values of DJ are the drug is more effective in killing cancer cells, and lower values are not so. It's not so. I mean, the drug is not working, right? And then we have a set of unlabeled data. Now I'm going to call that DI star, where this one is unknown on a set of patients where NI is equal to 1 to p. Okay. So the data construct consists of the known drug response for the cell lines, and the patients, and a set of an underlying CJ and PI is a high dimensional vector of genes. Okay, G genes. So conceptually think about 1,000 or on the order of thousands of genes on each of them, right? And what we are trying to get is how similar a given patient is to a given set of cell lines. And so to give you an idea about the dimensionality of the problem, so G here would be in the thousands, and the number of cell lines here, it's a few hundred, and the number of patients could be a few hundred to 1,000, right? And the key goal here is to infer the distribution of what a drug response could be on the given patient. And actually, I say drug response, but I should be a little bit more clear on that, or more specific in that. It's actually some level of therapeutic activity, because it can't be. We are not being prescriptive here in saying that this drug will work for this patient, but saying that this drug might have a higher therapeutic response, potentially have a higher therapeutic response, for a set of patients, because we are merging it with laboratory-based data, which in some sense is a much more cleaner version of the patient data, because the patients themselves are so heterogeneous, and a lot of factors contribute to their drug response. So it's more in the suggestive rather than the prescriptive domain, right? So our key here is the inferential goal here is to infer the distribution of DI star, which is the therapeutic index, and that we call it the Irx distribution. So for a given patient I, we want to get this distribution, and then given some model parameters, depending on how we parameterize the model. So that's the basic setup of the model that we want to get to, right? So I'll, and as I said, like if you have any questions, please feel free to ask in between. If there's somewhere I'm going on a tangent and some parts are not clear, I'll be happy to answer them.
So Veera, just one quick question. Yeah. For just DI or DJ, this universe, so you look at one drug at a time, or you actually have multiple drugs that you can sort of jointly model them?
Yeah, so right now we are modeling one drug at a time. And the reason we are doing this, and I'll get to how do we validate this, because we need a way to validate this, right? Like to say how well this drug works. So to validate this, I'll show you, to just give you a peek, we use existing trial data where we know the drug responses, but hide it from the model. And those clinical trials are only done one drug at a time. So if we get a good clinical trial, existing clinical trial data where they have tested multiple drugs, that will be a useful thing to do. But you could potentially build in multiple drugs and build in more dependence into this. But right now, just because that's the hardest part of this is the validation part of this model.
Thanks.
So for conceptually right now, you can just think about one drug.
Thanks.
Yeah. All right. So in defining this distribution, so as I said before, this is our data construct and this is our target distribution that we want to get to. Okay. So the way we derive this distribution is we want to get to the distribution of this therapeutic response given patients and some models. So it's just standard probability arguments. In defining a joint model on drugs and cell lines and conditioning on the patients, and just this is nothing super here, just plain algebra calculations. But what you can basically get down is defining a set of two models basically in defining this distribution. So it's a convolution of two models. One is the direct cell line model, where here the conditioning is the drug on the cell lines. So that becomes the training model, which is the prediction model. And then the cell lines and patients, cell lines given the patients, and that I call it the cell line patient model. And integrating with this with respect to C will give us this. So that's the breakdown of getting to this distribution by breaking it down into two sub pieces of the models and tying them together. And the major assumption of going from here to here in this, in having the math work out, is we assumed that this conditioning assumption, basically that given C and theta, C and P are independent. Okay, so the drug responses, C and P are not independent, but given drug response, given C and P, given C and theta is independent of P. And that's for two reasons. One reason is the theta here is taking care of that dependence between C and P. And the other is the way the data was collected, because the way the sampling was done, the cell lines were done in a different experiment based on the drug responses. So that sort of justifies making this assumption of doing this. So it's a reasonable assumption to make. Okay. So in essence, what this boils down to in defining this distribution is set of two distributions, and there is a lot of variety of choices one can take to define each of these two models to get at this distribution, right? And there's like this was a first pass at the problem. So we'll work with two Gaussian submodels, because then all the analytical expressions are analytically tractable. But in theory, it can be much more, one can complexify it much more using other techniques and for example, Bayesian nonparametrics or some sort of tools, more flexible tools, right? All right. So the bottom line here is we need to specify two sets of models. One is the drug cell line model, the supervised model, and the cell line patient model, which is the unsupervised model, right? So that basically then everything, once that setup is there, I think that's sort of the punchline of the talk, that one can define these two submodels. So for the drug cell line model, since it's a prediction model, we express it as a simple linear regression model, and in this case, what we'll pose it is actually a linear ridge regression model. But nothing stops us from being more general here. One can also do parametric models like any version of the shrinkage priors through lasso or horseshoe. Or even non-parametric, like trees or additive models using Gaussian processes or splines, kernels, anything under the sun. The reason we went with a linear ridge regression model is it actually works pretty well in practice, especially for prediction. That this is essentially becomes a prediction model, and it works reasonably well. And then we went with a linear model because then the computations are, the expressions are analytically tractable, and that just makes it easy to derive the operating characteristics of the model, especially from a validation standpoint. But if it's a well-calibrated model, once that is validated, one can build in more complex dependence, right? And just to mention here, we are still working with a high-dimensional setting, so the number of genes are much greater than the number of cell lines. So you have to use some sort of shrinkage or penalization here, right? So the key here is that we have a Gaussian error here, and that's needed to define a submodel that is Gaussian, right? So for the unsupervised model, so the way we do it is, first define a joint model of C and P together, these two subpopulations together, using a latent factor model. So a latent factor model is actually a dimension reduction technique, that breaks up, so basically C and P, if you stack them up, you have, an n plus m times P dimensional matrix, and breaking them up into two lower dimensional projections where you have a loading matrix and an F, and the lower dimension is indexed by a latent factor K. So this is a vanilla formulation of a latent factor model. And then we have the errors, but the errors have two components to it. So one component is for the patients, and one component is for the cell lines, and the other component is the patients. So that's phi1 and phi2. Okay. So essentially, these shared factors F capture the underlying genomic similarity using lower dimensional representations. And so in standard factor analysis, one takes K to be known, but in our estimation, we take K to be unknown. And I'll get to that, how do we estimate that? And so what this allows us is explicitly quantify the two source-specific variations. So these two are source-specific variations, phi1 and phi2. And this is the shared variation of patients and cell lines together. So basically you have the two matrices stacked one on top of each other. There's a set of shared variations, and then you have two source-specific variations. So these are the common, so this, you can think about the joint variation of these two is broken down into, three components. One is the shared variation, and essentially it will define three covariance matrices. And there's a lot of work in this area of defining source and shared specific variations. And Irina here in A&M, she's done a lot of work on this, and so has Eric Locke in Minnesota, and Gen Lee, who's now at Michigan, in understanding these sort of models, right? So the other nice thing of going with this parameterization of defining a joint model through latent factors on the cell lines and patients is the fact that this loading matrix can give us the dependence between the genes. These are the factors that load up on the P variables. So that can give you some idea about the dependency of the gene. And these latent factors F have a very nice interpretation of clustering. Okay, and looking at the latent factors and the loadings of the latent factors. So it actually serves a dual purpose. It sort of gives a dual, what is called a bidirectional clustering of both the rows and the columns. So if you think about the columns to be the genes and the rows to be the samples, so the A gives you the column specific clustering. So that's the genes. And then the Fs gives you the row specific clustering, which is which is useful to, as I'll get to, in defining the similarity between cell lines and patients. So what are the best cell lines that match to a patient, right? And that's useful as well. So basically the justification of using a latent factor model is just the ease of interpretation and the byproducts one can get out of a latent factor model, right? Right. So once you have defined a joint model on C and P together, now one can write down it's a standard it's pretty straightforward actually to write down the explicit factorization of the cell line patient model of C given the shared variation, P given the shared variation. Those are two Gaussian submodels. And then and tying that with the drug cell line model here, and doing this integration with two Gaussian submodels, it actually turns out that the derivation of this D given P, and actually there should be a theta in here, is a Gaussian distribution. Okay, so basically that's the argument of using two Gaussian distributions here, that you can get this explicitly. And the reason we wanted to get this explicitly, this distribution as a first pass, is how well, we just didn't want to get one number of the drug response. What we wanted to get is a distribution of the therapeutic index, so that this whole distribution can be used to calibrate the therapeutic index. Right? And then once you have the distribution now, and especially once you've gone through the Bayesian crank, once you get the distribution, now one can use any distributional summary to compute the actual score that one wants to use. Okay, and that could be the mean, variance, or any quantile of the distribution, right? And if you have multiple distributional summaries available, the easiest one to take is actually the expected value. So to define the theoretical individualized therapeutic index, we call that the Irx score. We take the expected value of that distribution, right? And that's the theoretical expression of the expected value of that distribution. But in practice, once you have estimated the model parameters, you can actually derive it for each patient. What is their therapeutic index, and that just we call it Irx score. All right. So if you put in all the data for C, P, and the drugs inside it, what will end up is this distribution, and from that distribution, the expected value of the distribution will turn out to be this distribution. And the key thing here is that this pops out as a term with the AA transpose, and that's the dependence on the shared variation. So I guess the next question is like, why go through this elaborate procedure, and what does the shared variation buy you, in a practical sense? So if you compare it to a naive approach, as I mentioned before, where you use a different training model and a different test model, and do it as an independent, so that we call as a naive score, where you basically just take the drug cell line model or just the training model and plug in the estimates, right? Actually, we can theoretically show that the prediction error under a shared dependence will always be less than the prediction error under an independent model. All right. So to be honest, we actually fell back on this. We fell backwards into this claim. We actually did the simulations, and the simulations were, to ask the question like, what does shared variation buy you? How much does that buy you? Like if you indeed have a shared variation, should you get better prediction? And we found that the answer was yes, that in a variety, and I'll show you a gist of those results, that if you have shared variation, you get better prediction. But to take it apart and see, why do we get an increase in the prediction? Why do we get a gain in prediction accuracy or some level of efficiency? And that comes from the fact that the difference between the two turns out to be a quantity, which depending on the amount of shared variation you have, it's a non-negative quantity. And that's a nice, I wouldn't say it's a theoretical argument, but it's more, an operating characteristic argument that you see it in the simulations, and why does it gain? And that's sort of the argument of why it comes through. All right. So the bottom line here is, you get better predictive accuracy compared to just plug-in approaches, where you don't, borrow strength across the two data sources. And you can, under a Gaussian submodels, you can actually explicitly quantify the accuracy gain. And that's sort of reflected on the simulations as well. right? Right. So, so I haven't talked about priors at all. So, as I mentioned, I might have given out it before as well. Like we turned the Bayesian crank on this one. So we basically have priors for each of the model parameters. So as I said, the drug cell line model, we kept it really simple and used a rich penalization, and as just standard Gaussian models with beta following a rich penalty with the standard gamma distributions. And this latent factor model, and we punted on this a lot, like what is the best way to estimate? And we hit upon this actually really nice paper by Bhattacharya and Dunson that they propose what is called a multiplicative gamma process, an MGP prior. And the nice thing about the MGP prior is a very parametrically tractable prior, and where you can also determine where you can keep the number of latent factors unknown and then estimate it using an MCMC approach. Okay, so just to delve into a little bit more, essentially, it's like a scale mixture of normals, where the loading matrix you have so for any generic loading matrix A, on each element there is a Gaussian prior with two variance components. And then one of the variance components follows a gamma prior, and the other one is sort of like a convolution of gamma distributions, hierarchical gamma distributions, and that induces. So what they showed in in their paper, and we found out in our paper is in our implementation rather, not we didn't reinvent this in any form, is that that gives really nice shrinkage as well as estimation of the number of shared. Okay. So once you have a process latent factor process prior on the supervised part of the model, and combined with that, one can do all the posterior computations. It's pretty straightforward, to be honest. That and also doesn't require that much of tuning, in calibrating that. Right. So. Right. So I'll just do a quick pause before getting to the simulation and the results, if there are any questions on the model. I guess I'll take that as a now, and then if you have any questions, as I said before, please feel free to chime in. All right. So the simulations, we did a bunch of simulations, and the main idea of the simulations, as I said before, was to empirically validate the gain in prediction accuracy under a joint model versus an independent model, and then assess the impact of the source specific variations. So, if you have two sources of data that are completely different from each other, should you see any gain in accuracy, as compared to the case where there is shared variation, and how much gain in efficiency. So for this simulations, we just calibrated the whole simulations based on the real data, where we had around 300 cell lines and 200 patients, and we took around 1,000 genes and real data. Okay, so here's like one of the simulation settings that I'm showing you here, and what we did here was we had a fixed amount of shared variation that we got from the real data. And then what we did was we changed the levels of shared variation. So in this case, we fixed the cell line variation as fixed, and then changed the patient level variance, the patient level variance source. And in the other case, we kept the cell lines the constant, and we changed we kept the patients constant and changed the cell lines. And the bottom line, what we saw is that the calibrated model, does much better than the naive prediction model in all scenarios. And that, as I said before, is attributable to the shared variation. So the more shared variation you have, the better you will do in terms of prediction accuracy. And that's the idea. Okay. So this was the hardest part actually of the whole exercise, was how do we actually do clinical validation? You get a therapeutic index and you say, okay, this drug is going to work, but how do you actually in practice say this is a good one to use now. So so the hypothesis is, is again, how do we, to just dialing back to the original questions I had asked, is how does pulling information across labeled and unlabeled data improve calibration accuracy, and how do we validate this in a drug response for therapeutic drug response. And sort of the two byproduct questions we also wanted to get answered is, what are the optimal avatars of cell lines for patients, and what are the genomic factors that are driving this? So these were the scientific questions. So for this analysis, we took a system of cell lines from what is called a genomic data. I forget what GDSC stands for. It's a pretty standard database for cell line models. They have a comprehensive data from multiple cancers. And this log IC50 is basically the drug response. As I said before, it's the amount of drug that kills 50% of the cells. So that's a continuous response. Okay. And then we searched actually a lot to find the right clinical trial to apply this on. So we found two clinical trials, multiple myeloma and breast cancer, where they were studying this drug called bortezomib in multiple myeloma, which is basically a sort of a proteasome inhibitor that's given to advanced stage multiple myeloma. It's sort of like a blood cancer, and it's actually become a standard of care right now. And finding that, who would be responders. So the responders, if you give a drug, they have some level of complete response, partial response, or I forget what MR is, but some level of response versus having no response. Right. So in essence, we know, given this clinical trial was already done, this was done back in 2007. So what we have the data is the response, as well as the genomic features, okay? And this response, we mastered for validation. So basically that didn't enter a model fitting at all, okay? So that is so it's in some sense, it's a pure validation that we did, not a cross validation. It's a pure independent validation. And then we found another trial in breast cancer that does docetaxel it's a much smaller trial, about 24 patients, that had response and non-response defined by some level of tumor shrinkage that they did. Okay, and so we used so both of these trials collected gene expression on these patients. That's the hard part, like having trials that collect both genomic data and drug response. Those are hard to get, but they're maturing pretty well. So hopefully we'll get much more data sets to more thoroughly validate it and build more on this. And using the gene expression data, after some pre-processing and normalization, we had around 1,000 genes that we used for fitting. Right. So here this is essentially the main result. Is what I'm showing you here is box plot, the violent plots rather, of the predicted therapeutic index for the non-responders versus the responders. Okay. And you can see it's not like a pie in the sky results that you have a big difference between the two, in terms of the entire distribution. But if you do a simple, t-test between the responses between the two groups, under our model, we get some level of significance. But if you run it under a naive model, you don't get any significance. And similarly for this breast cancer data sets, that you see a separation in the therapeutic index, and also that is in the right direction. That means higher therapeutic index means that drug, in this case will not work, will work rather, sorry, the lower therapeutic index. So in this case, it's lower in the responders and in the non-responders, and similarly here. So it's in the right direction, and it provides some amount of validation. I won't say it's a perfect validation, but at least some amount of validation that it tends to do better than naive approaches, right? And as I said before, this can also be used to define the avatars of the cell line avatars of different patients. So just to briefly skim through, what is of importance is with the latent factors, you can do the clustering as well, and then the important clusters are the one where you get a mixtures of patients and cell lines. Those are the more like, as opposed to clusters that have only patients or cell lines. And in this case, there are 3 clusters that showed a clear mixing between them. And most of these clusters are, so shown here as well by a chord diagram. So the responders completely, most of them go towards these set of cell lines. So these are sort of the representative, cell lines for the responders, and these are the representatives for non-responders. So it gives some not intuition, but some evidence of how to choose the best cell lines to study further in the context of this drug, or in the context of this cancer, right? And also, as I said, like you can use the loadings on the A to define the relevant genes, and then we did some further investigation of those genes and pathways, and they have some biological justification in terms of the multiple myeloma. In terms of their role in multiple myeloma. So there's some additional biological interpretations about these pathways. All right. So Yang, how much time do I have?
8 minutes.
8 minutes. Okay.
Yeah.
So probably on the network stuff, I'll probably just give a brief intuition. I'll just give you the flavor of the problem rather than going into the full technical details. So this was a related approach where we took a network-oriented approach to this problem, where like basically networks, the definition of the intuition behind networks is to having a more systems level overview of these. I'll probably show a picture that would make much more sense here. Yeah. So basically, what we wanted to develop is multi-layered networks where you have, different sets of variables at different layers. So if you think about the first layer having a DNA level layer, which is the DNA level data, and the second level being an mRNA level data, and the third level being a protein level data, and tied it with a clinical outcome. Any outcome of interest, that could be a drug response outcome or any other outcome, and understanding these relationships, both within layer and also between layer, and building these networks. So this goes more into the mechanism, so if there is a specific biological mechanisms that, which are within layer specific or across layer specific. And this, we call it a multi-layered network, multi-layered genomic networks. And this is a paper that actually just got published a few months ago in April in JASA. And Minjin Ha was the leader on this work, along with actually one of my other ex-colleagues, Francesco Stingo, that we worked on, right? So it actually turns out this is a cute problem in its own right, that defining networks, defining networks across layers, where the layers have an ordered domain. So if you think about, you have Q sets of variables, which are the layers, and you have an ordered domain. And what you want to get is sort of the dependent structure, which might be across layers, which are shown by arrows here. So they have a directed relationship, but within layers, they have an undirected relationship. And you could have multiple layers, and a nice, parameterization or a nice conceptual, framework for this is what is called usually chain graphs. So it's basically building a chain because you have a layer specific network. So what we essentially did was, it's defining a joint graphical models across these whole sets of layers, together to get inference on both these undirected edges, also with directed edges. And this problem is, when you think about, multiple number of genes and multiple number of layers, it actually becomes a very high dimensional graph network estimation problem. So our key contribution to this was actually defining what we call a bands framework, where you can do actually reduce the whole joint distribution into a bunch of regressions, univariate regressions, and how you can tie up the univariate regression to get to some version of the joint, not explicitly, but at least in empirical performance, it can get to the joint distribution, and because so that helps one computationally, but also in terms of specifying priors and such. One can specify a rich class of priors when you reduce it down to a regression setting, because that's a well formulated problem, and one can do that. So that was basically the essence of this work, and I'll be happy to talk offline or send references on this, and of course, like, we did a lot of simulations and real data analysis in pan-cancer omics, in different types of cancers, to both illustrate and the practical, but also the operating characteristics of this method. So I'm going to leave some room for questions. So I'm going to whiz through these slides, but I just want to mention a couple more points here. One is actually this model, as we thought more about it, this has actually a nice interpretation in the setting of causal analysis, which we didn't, this connection, we didn't make it in the original paper, but this is something we are just actually thinking through at this point, is, and this gets into actually mediation analysis, specifically within the context of causal analysis. Is if you think about mediation analysis, you have a set of mediators. Usually, there are single mediators, but in the case of multiple mediators, and you have multiple exposures, and you have a set of covariates. So under certain assumptions of covariates, exposure, and mediators, you can actually derive causal interpretations from these multi-layered networks as well. And that's something we just starting to explore at this point, right? And so I'll skip through this. We have extended this to multiple other cancers and data types. This is a work by one of my students, Rupam Bhattacharya, that came out in Journal of Clinical Oncology, that we are extending this to. And so I just want to mention that we have a lot of resources on our GitHub page on all the codes and software for this. So that not only includes the Shiny apps that come from this work, so all the results that come through Shiny apps, and actually my students who are much more involved than I am, and they actually made videos of how to use that software. So I have a couple of students who actually have started a group YouTube channel where they put in this couple of minutes videos about how to actually use this software from a practical standpoint, which I thought was brilliant to further enhance their work and the impact. Right. So just in closing, like I started with a very specific example in precision medicine, in precision oncology, but there's more general methodological uses in other scientific domains of this calibration problem when you have two sources of data where one of them is a labeled and another one is unlabeled, and there could be more potential broader uses. So right now we base the whole model on linear and Gaussian assumptions, and that was, as I said before, is to make the tractable mathematical computation analysis, but also in terms of implementation. But something we're thinking is how to robustify that through potentially non-parametric Bayes approaches. And there are some theoretical aspects of this sort of missing data approach that we would like to investigate a little bit more. What is the level of missing? Because this is in some sense a complete missing data of the responses and those and sort of get through those. And there are a few other methodology application specific extensions that we're still trying to figure out, is in terms of improving, like one of them that Yang mentioned is looking at more drugs, incorporate multi-omic data. We only considered expression data, and so on and so forth. Right. And I've already attributed who all did the work on this, and then hopefully Scott, hopefully I haven't confused you enough. No, if you aren't convinced, at least I have you all confused. So I'll stop there. Thank you.
Okay, thank you very much for the very interesting and informative talk. So I guess we have a time for one or two quick questions. Do you have any question from the audience? Well, let me first ask a quick question about the real data. So for the clinical trial data, is the genomic data collect after the drug or before? Is it the baseline genomic data, or is actually the genomic is measured after the drug has been applied to the treatment group?
I think it's baseline. Most genomic, yeah, because it's it is baseline because they do the tumor staging and the biopsy before, right? And they keep some of the material. Like I'm 99.9% sure it's baseline, because usually after drug it's harder to because then it's a confounded effect, right?
Yeah.
That you can't parse out that is it due to the drug or was it already there. Right. The whole point of doing this is you want to get it at the baseline.
Yeah, so that you can predict which one, whether the drug is going to work for this patient or not, based on the index you proposed. Yeah. Any other questions for anyone?
I had a quick question if there is still time here.
Sure.
Yeah.
Hi, Veera. Very nice talk. Sorry, I don't have video currently, but a very nice talk. And
thank you.
I just wanted to ask you a couple of quick questions about the semi-supervised setup that you have mentioned. So generally in semi-supervised, one issue that people often encounter is the unlabeled data is often much bigger than the labeled data. Is that something you encounter here? And the related question would be, because this is very much a missing data problem with responses missing essentially, right? So do you have a situation where it is missing at random, like the labeled and unlabeled data can possibly be differentially distributed, versus are you actually just assuming they're equally distributed so that it's more like missing completely at random and the question is solely about leveraging the unlabeled information to improve efficiency as opposed to like ensuring consistency?
Yeah. So both are really good questions. So let me say the first one. This is actually the reverse case here. In the first, like when you said the size of the labeled and the unlabeled data. So the size of the labeled data is much less than the size of the unlabeled data. Because, the patients, the consumption level effects. So if I think just cancer, for example, this specific case, we have genomic data on thousands up to 10,000 cancer patients. But the cell lines, these studies to evaluate drug sensitivity, these are very expensive cell line studies, drug perturbation studies. So you have
right. So that's what I was sort of hinting at. It's kind of like an extreme version of a missing data problem, right? So
that's right. So yeah. So the second part of your question, to tell is it missing at random? What is the missingness mechanism? To be honest, I don't know, because it's not that these patients went to a clinical trial and the response was not taken for some reason. It's just not that the experiments haven't been done yet. No. So what we are trying to calibrate is, for a certain cancer, if you have a given drug that's worked really well on cell lines, and you want to take it to the clinic, we have the genomic data from these biobanks. Can we use that to sort of prioritize them? It might be that it's not the drug responses in the absolute, but you could get a relative ranking between the drugs. So essentially,
right. But I suppose my thinking was whether this database on the genomic features, the target population for that is somehow any different from the target population that you are actually sort of getting from the label data. That's essentially what would mean that they are different distributions essentially, and MAR type of an assumption rather than MCAR.
Right. So the underlying assumption is there should be some intersection of that distribution, right? And that's the assumption that folks usually make when they develop drugs, right? Like they say, okay, it works on cell lines. We're going to take it to, mice, and then we're going to take it to humans, right? And so the cell lines are sort of some representative versions of humans. How representative, that that's an unanswered question. So there could be better forms of the labeled data that one can work with. And I didn't get to it much. Actually, one of the projects that I'm working with is getting better sources of data which have labels. And one of them is what is called patient-derived xenographs. So it's essentially a patient walks in, they take the cells from that patient and grow it into a mice and do an actual clinical trial on a mice. And then that's the hope. So the idea is, what is the big picture vision of this? When I was talking to my collaborator, what they want to do is they have a DVD of this, like a collection of these cell lines, and then a patient walks into the clinic, they can just pick, say, okay, this is the best match. And I'm going to do preclinical testing only on these cell lines instead of, just arbitrarily choosing them, giving them a guided way of doing that.
I see. Okay. Thanks a lot, Veera.
Yeah, sure.
So sorry, for the interest of time, let's thank our speaker again, because Veera has a meeting with Anirban actually right now. Okay, thank you very much.
All right. And please feel free to contact me if you have any questions, and I'll be happy to, answer them or send references along.
All right. Thank you.
Thanks.
Yeah, thanks a lot.
Thanks, everyone. Bye-bye.